Hello friends, welcome to my video. In this video, we are going to see about bacterial genetics. Before saying what is bacterial genetics, we are going to see what is genetics. Genetics is a branch of biology that deals with the study of the heredity and the variation of organisms. Next, we are going to see what is heredity. Heredity is the passing on or the exert transmission of genetic information from parents to their offspring. Heredity is also known as inheritance. Next, we are going to see what is variation. Variation is the presence of differences in sequences of genes between individual organisms of a species. Mutation, sexual reproduction, and genetic drift are the sources of genetic variation. Thus, genetics is the study of the inheritance and the variability of the characteristics of an organism. We learnt what is genetics. Next, we are going to see what is bacterial genetics. Bacterial genetics is the study of the inheritance and the variations of the traits or the characteristics in bacteria, which means bacterial genetics is the study of how specific characteristics or informations is transferred from a particular bacterium to its offsprings and how it is expressed. Next, we are going to see what is bacterial chromosome. Most bacteria have a haploid genome, that is a single chromosome, and the bacterial chromosome consists of a double-stranded DNA molecule arranged in a circular form. It is about 1000 micrometer in length. The length of DNA is usually expressed as kilobases. The bacterial DNA is about 4000 kilobase long. You should note one important point here, that is, usually in bacteria, single circular chromosome is present, but exceptions are there, where some bacteria may contain two chromosomes, and in some bacteria, the DNA is linear rather than circular. Next, we are going to see about DNA. DNA is the genetic material carrying the code of life. The DNA molecule is composed of two chains of nucleotides wound together in the form of a double helix. Each chain has a backbone of deoxyribose and phosphate residues arranged alternately. Attached to each deoxyribose is one of four nitrogenous bases. The four types of nitrogenous bases in DNA are adenine, guanine, thymine, and cytosine. Adenine and guanine are double-ringed purines. Cytosine and thymine are smaller, single-ringed pyrimidines. DNA bases pairs up with each other to form units called base pairs. The sequencing of this bonding is specific. That is, adenine bonds only with thymine and cytosine bonds only with guanine. Hydrogen bond is present between the bases on the opposite strands. Next, we are going to see about DNA replication. The DNA molecules replicates by first unwinding at one end to form a fork. Each strand of the fork acting as a template for the synthesis of a complementary strand, with which it then forms a double helix. Next, we are going to see what is semi-conservative mode of replication. During replication of the bacterial genome, each strand in double helical DNA serves as a template for synthesis of a new complementary strand. Each daughter double-stranded DNA molecule thus contains one old polynucleotide strand and one newly synthesized strand. This type of DNA replication is called semi-conservative. DNA acts as a template for the synthesis of mRNA. Genetic information is stored in the DNA as a code consisting of a sequence of 3 bases. That is, the code is triplet. Each triplet transcribed on mRNA determine a single amino acid. Next, we are going to see what is a genome. An organism's complete set of DNA is called its genome. The study of genome is called genomics. Most bacteria have a haploid genome, that is, a single chromosome consisting of circular double-stranded DNA molecule. Next, we are going to see about genes. Genes are a segment of DNA or sequences of nucleotides within DNA that code for gene product, either RNA or protein. I'm concluding this video here and see you soon in my next video. Thank you, friends.